So for disinfecting performance, we look at the International Standards Organization and Food and Drug Administration standalone procedures as standards for disinfecting efficacy of products. We know that there are 5 panel microorganisms that are used in the standard, 2 bacteria of a gram-negative variety, 1 gram-positive bacteria, and 2 representatives of fungal microorganisms. And of course, the standard is a 3-log reduction for the bacteria and a 1-log reduction as minimum performance for the fungus. If we compare these market-successful products in their disinfecting performance, one of the things that we'll notice here is that they all meet or exceed the standard because they needed to, of course, in order to be marketed to the public. We see here a very strong performance by Renew MultiPlus, well exceeding the standard for each one of these panel microorganisms. We also see that OptiFree Express exceeded the standard. OptiFree Replenish shows a strong performance as well, and Complete Moisture Plus, in addition, is one of the market successful products out there that we are comparing here today. The 5 panel microorganisms are representative of the diverse microbial challenge that one might encounter in the real world. And in fact, we know that there are different strains of Pseudomonas, different strains of Fusarium, Staph aureus, and so forth. So these panel microorganisms are very particular American type culture collection is what they're called, an ATCC strain, so that anyone doing this testing anywhere in the world or doing repeat testing will always use the same strain of the microorganism as the representative ones for these performance testings. So that's what's required. But what we've learned of late is that when we look at real-world situations, patients will encounter different genetic variants of these microorganisms and different strains. And so what we can do is look at clinical isolates. Those are microorganisms that are recovered from patients who've had an eye infection. It's either recovered from the eye itself, from the contact lens, from the contact lens case. And so the genetic makeup of these other microorganisms may very well be different than the ATCC strains. They've mutated, they've been modified, and of course the pharmaceutical industry is always trying to come up with newer and better antibiotics to kind of chase after the adaptations that these microorganisms make out in the real world. So there's some value when we compare disinfecting performance profile of these lens care products to looking at their performance against clinical isolates from the real world. Now, realize that using these test protocols is good because it's the standard. We use the same ones, but test against clinical isolates. This is not required for product market approval by the FDA. But doing so merely illustrates the performance of the leading lens care products against these real-world variations of the microorganisms. So it does give us some insight on how the disinfecting profile might be a little bit different from these leading care products against clinical isolates. So an example of 11 Staph aureus infection-causing clinical isolates subjected to these leading lens care products. We see that Renew MultiPlus exceeds the standard. It's more than a 3-log unit reduction of these clinical isolates. The other thing that's important to recognize, it's a very consistent performer because the range of disinfection is quite tight. So a very consistent performer. Another PHMB-based product, Complete, is also exceeding the standard with a tight range of performance. OptiFree Express does exceed the 3-log unit standard, although the range does dip down below that standard. And OptiFree Replenish, with its different formulation, actually doesn't meet the standard. There's not enough log reduction in these clinical isolates of Staph aureus. And in fact, the range is quite wide in its performance, and only at the top end of the range does it seem to just barely exceed that 3-log unit reduction. So again, these are all marketed products, very successful out in the real world, and yet when we look at the disinfecting profile against some real-world clinical isolates, we do see some differences here in their disinfecting performance. there are genetically different fusarium microorganisms out there. In this example, 20 real-world clinical isolates from Fusarium solani infections. We see that the robust nature and the effectiveness of Renew Multiplus is not exceeded by any of the other products. It well exceeds that 1 log unit reduction in these clinical isolates with a moderate range of performance. OptiFree Express also exceeds the standard. The complete lens care product does as well, and OptiFree Replenish is also effective against these real-world clinical isolates. So the good news is that while there are differences in the robustness of these disinfecting solutions against these clinical isolates, they all do exceed the standard. So it's all very good news there. The other question that we might have from the recent past, episodes of infection with patients using lens care products, is what are the effects of non-compliant conditions on the performance of these lens care products for patients out in use in the real world. So again, using the same standard testing protocols that are used for passage and approval of these lens care products with the ATCC strains, maybe we can use those same protocols under extreme condition testing. Now again, this is not required for product market approval by the FDA, but doing this kind of testing would merely illustrate the effects of some known forms of patient noncompliance and are used as a benchmark against some of these accepted standards. So noncompliance. Examples of noncompliant behavior. Not cleaning a lens case regularly is a very common thing, or perhaps not replacing the case regularly so that they get quite dirty and a source of potential infection. Poor cleanliness with the caps and the tips of the lens care product bottles. Opening the bottle caps or the lens cases and leaving them open is another potential problem. Basic hygiene of not washing your hands effectively or incomplete rinsing of your hands before handling lenses. Perhaps not rinsing the contact lens, and something that I think is quite common, and that is topping off the solution. Instead of emptying the case of the old solution and starting over with a full supply of fresh solution in the lens case well, the tendency can be to just go ahead and add to what was there and top off. So given these non-compliant behaviors, how do we add this element of non-compliance to our testing procedures to look at disinfecting performance? Well, simulating non-compliant conditions of leaving a lens case or a product bottle open is one way to do that. And we know that if there is no evaporation, testing against clinical isolates of fusarium under these conditions of simulated non-compliance, we'd start with no evaporation. We see that there are differences in the performance between these products, but they do exceed the standard of 1 log unit reduction. Extreme conditions of water loss where we have a concentrating of the other components in the lens care product throws the balance out. It changes what these product formulations were meant to be. And we can see with 50% water loss and even a very extreme condition of 75% water loss that there are differences in how these products perform in their disinfecting capability against different clinical isolates of fusarium. In particular, I'd point out that the OptiFree Replenish does lose some effectiveness after 50% evaporation. So again, these are accepted, product-marketed, successful products that are out there. That's the good news, is that they really do work. But again, if we look at these extreme testing conditions not required for approvals for marketing, we do see that there are some differences in the disinfecting performance profile.